Today I will be talking about the major problem we have been seeing right here sitting Dr. Subhranshu Chatterjee and I a year back was discussing about that multi-drug resistance is a big issue into any field you go, is it whether it in human or we can say in animal husbandry. So priorly talking about the antibiotic quanta, I will prefer to talk about our new innovation called Fly Seal, which can help the poultry field and the industry to see that the chemical industries can also do some of the innovation and give a better result, which already existing innovations which are been there into the humans. Like one is estradiol phosphate, which is the exact opposition you can say to Viagra. Right. So larva, if we can consider, then firstly, when larva has been converted into the fly, there are the many stages. So if we potent them, that in very, you can say commercial language, you can say that if we potent them into the parts and their private parts do not work, then what will happen? Larva will not convert, or the end fly cannot make the larva themselves. So these are the reports which we have created with the help of my in-house scientists. We have been working on estradiol phosphate, which is one of the major hormones into the females. Actually, estradiol phosphate is given into the humans for controlling the birth. Right. Here you can see the data is the genetic data of the fly after giving Fly Seal. This powder was being spread onto the larva, and what we have observed is that when this larva is converted to the fly, these are incapable of reproducing another flies. Please go down. So here you can see these are the normal conditions where the reproductive parts have been damaged. Fly itself is fine. The larva is converted into the fly, but the fly then cannot convert the another larvas. It is incapable to do that. The reproductive system is totally damaged. So now this can be the actual solution, a big solution to the poultry industry, that the larva is been not formed from the existing flies. It may happen that the new fly can come, they reproduce, but if you will spray again this, then the reproductive parts have been damaged. So now, estradiol phosphate, which is a very commercial product into the humans, you can say, we in day-to-day life, we are using it, and we were manufacturing estradiol phosphate from last decade. So we have observed that if we can include this into the poultry industry and convert it into a different product, it will be a very better achievement. Please, can we go down? Yeah. These are also some of the another results, which are the blood reports after the fly, means the flies which have been given estradiol phosphate, that fly were given to the birds. We have made that fly eaten by the birds. So what happened is that the blood reports and everything is normal. Their reproductive organism has been not changed because it is in very small quantities. If we can say that a cycle of a small baby fly and the cycle of this big animal, mammal, is totally different. So it has been doing no change into the reproductive system of the poultry itself. This is the effect on the larva. You can see that the control which we have seen where it is normal, 20 mg per liter, the dose is given 10, 20 and 40. If you see 40 mg per liter, the total damage of the reproductive system has been seen. Now this fly is incapable because the estradiol phosphate has affected the biofilm of that fly. If you can see these graphs downwards, the first graph and the last graph, if you see, has the major difference. From the 100% potency, it has given to the 10%. So 90% of the fly control you can be do just by giving estradiol phosphate into the larva. So these were the results of the fly cells which we actually got right now. So I gave him to present it. Yes. So majorly for what I came here is an antibiotic which we found. This is a novel scaffold which we have developed. Now, if we go to the antibiotics right now into the poultry, what is happening is that the generations of antibiotics has not changed, but the bacterial generations have changed. If you see, then third generation bacteria are been developed. Now, multi-drug resistance is there also into the humans, but many drugs are available. You can say combinational drugs are also given. Scaffolding is also done into the humans for a new development is also done. But as priorly when I was been here in morning, we are saying that there is no novel drug or another drug into the poultry from many decades. There is no another antibiotic seen into the poultry. We require a new molecule not just because the MDR, but also because that the older antibiotic dosage are so much that the body of the poultry, when it ejaculates the from the biofilms when the antibiotics is ejaculated, some amount of antibiotic still remains into them. And what happens is that that antibiotic is being eaten by humans. So lesser the quantities will also give the make big difference into the poultry as well as into the industry. Now, why do we need the multi-drug resistant drugs? Firstly, that if you see the second generation and the third generation difference of the bacteria itself, these are the common antibiotics right now used into the poultry. If you see that priorly it is affecting, but when we change the generation of the bacteria, it has no effect on it. So what happens is that we increase the antibiotical dose. If we are giving, for example, 30 gram per ton, now we are giving 200 gram per ton. So what is happening is that we are increasing the dose of the antibiotic, but not changing them. The main thing we need to do is to change the antibiotic, which can give the good resistance and work well to the resistant bacteria. The antibiotic name is Quanta, and we have done some results on bacteria as well as the fungus. You can see these are the strains of bacteria we have taken and the results on them. This is the zone of inhibition in which 52 mm, 50 mm and 60 mm of zone inhibition is seen, which is the best results you can get into an antibiotic. Now minimum inhibitory concentration MIC value of this drug if we talk, then that starts from 0.01% to 2%. Means 0.005% is also working on the bacteria. This same concentration also goes for fungus. Like you can see, these are the results of fungus. If it can kill fungus, then the same concentration is capable enough to kill the bacteria also.
Mr. Punit, I would like to interrupt you for a moment. I would request here, everyone, please keep your mobile phones on silent and not talk among ourselves so that it is easier for the speaker to interact. Thank you so much.
Yeah, thank you. So this is the certificate of analysis of that chemical. How does it look like? And what are the major descriptions of it? It is soluble in acidic and basic media. So main reason beside the solubility is that there are different kind of effects of antibiotics. How, in where it is dissolved. Is it dissolved in stomach? Then if it is dissolved in acid, then what will happen? It will be getting more effect towards the intestines and then into the colon. So this is soluble into the stomach itself. So it will give very good results onto the intestine and after that to the colon, which gives the gram-positive and gram-negative bacteria both into the contact and give very good results for the antibacterial effect. We have done acute toxicity also of the molecule, some of the results on it, which we have done on rat, and dose also is written. You can take 30 to 100 gram of antibiotic in 1 ton feed. Antibiotic has the target. So what is we targeting on? How this antibiotic works biologically? What is the biological mechanism of this antibiotic? That is very important to understand. So these are the novel targets for any bacteria to kill. The main fundamenta is to kill the bacteria or kill the virus. So we are focusing on ATP PRTase. Now what is ATP PRTase and how the target has been working? PRPP and ATP, these are the two targets of any bacteria. In presence of magnesium, they react. When they react in presence of magnesium, two byproducts internally is formed, which shows into PPI and PRATP. This PI and PRATP and PRPP and ATP, it is an irreversible and reversible process both into the body in presence of magnesium. Now what we are doing is, when it is converted into PRATP convert into L-histidine, which is a major amino acid for any bacteria to sustain the life. If we can stop conversion of L-histidine, then it is possible to kill any bacteria. It will not be alive because L-histidine formation has been not done. So targeting ATP PRTase is very new and novel synthesis are been done. This scaffolding is also been done by the crystal structures also. The crystal structure which you see here contains an molecule inside. This is our molecule which you can see in number C and D. This is an overall 3D structure of the whole protein which we are targeting on. And these are the HyBiz inhibitors right now found. Means in 1976, these 3 molecules HyBiz inhibitors were been found. In 2008, these 4 HyBiz inhibitors were been found. In 2009, this HyBiz inhibitor was found. And last HyBiz inhibitor which we have found was in 2012. After that, many human antibiotics have adopted this and HyBiz molecules are being used. This 2012 molecule is being marketed by Sanofi. You can say this is marketed by Sanofi right now also in urinary tract infection. This is our novel targeting and this is the MIC value of the molecule. Now, if you see the scaffolding of any chemical structures, there are many kind of scaffolding like benzoimidazole, benzothiazole, benzoxazoles. These all scaffolding are being done on the base of what target you have. And this all, you can say till 2018, HyBiz was the main target for any pharmaceutical company to contemplate the antibiotic to form. Majorly, the irreversible process of resistance has been a big challenge for humans also. So if we can take this as a point and make this into the animals also, that if the resistance of the bacteria can be converted towards the irreversible process and we can kill them, that is a big opportunity here to any antibiotic we can form. These are the aza-indoles and indole scaffolds which we have developed. These are the patented molecules by us. The MIC value is 0.0195 μm. Another is showing 0.013 μm, 2 μm, 0.12 μg. These are all designing which we have done from converting to 0.0009 μm to 100 μg per ml. So this is how we have come to the development of the molecule. We have taken the existing drug molecules, taken 2 parts of that existing drug molecules and converted it into the one molecule. So the two antibiotic which is existingly been used into human, we have taken the scaffolds of two drugs, converted it into the one drug, and then seen the results, which is really fantastic. This is the CADD, in which you can see that computer docking has a score of minus 9.3. If you will give this data to any pharmaceutical company, they will be astonished that a molecule can give docking score of minus 9.3. This is a really big achievement. I will say that Dr. Subhrangshu has done a very great job with us in development of this antibiotic. We have been working for 3 years and we have achieved this great thing that a molecule can give minus 9.3 docking score. This docking score means that it is very potent drug which can kill any existing antibacterial or bacterial or viral or fungal infections. So these are all scaffolding which we have done, and this side you can see the amino acid interactions also, that this drug is joining which amino acids. The essential amino acids which are present here, you can see that these all are the amino acids which this drug is targeting on. And this is the 3D structure inside the body. When the drug is given inside the body, where it gets joined, the His-G inhibition and the interaction of amino acids. With this amino acid, this molecule has interacted. You can see here. So what are the objectives actually to how to develop this antibiotic and accept this as a novel indole 2 carboxylate as the novel usage for giving the farmers an opportunity to take this multi-drug resistance into the daily cyclings. So we are even capable enough to synthesize this molecule. We have been synthesizing this molecule approximately of 100 tons per month. Today, if we can talk, while talking I am synthesizing this molecule 100 tons per month. And we have been larger distributors to Syngenta also, in which we have been manufacturing many of the herbicides. So the molecule which you see is a commercialized product right now in European market, as well as you can commercialize it into the India. This drug has affordability, means it comes with the price range of 1350 only, 1350 per kg. And the multi-drug resistance of this product gives you the assurity that when the lower dose of this molecule will be given to the animal, the end result of this molecule will be fantastic. You will not see any damage done by the bacteria or fungal infection into the animal. The bird will be free with the fungal and bacterial infections. Also, the dosage is so low that you can say that after conversion and biological process has been completed, the minimum amount of antibiotic is going to the humans, which is the major problem you can say into the poultry right now. The big, you can say, issue or problem arised by the government is that how antibiotics has been taken into the poultry industry, and then that antibiotic has been consumed by the humans also. But the main thing is that after the completion of the biological cycle of any antibiotic or any molecule, it is excreted out by the animals also. So when 7 days to 10 days is completed, the excretion of whole antibiotic has been done. But the main issue is that the antibiotic which is given is in how much amount. The excretion time also depends on the amount. If you are giving 200 mg, 500 mg to 600 mg to 1 ton, then that will definitely cause the more amount of time to excrete from the bird also. Lesser the amount, more the efficiency, greater the results. That should be the motto. These are the NMR and mass data which we have done, which gives the 100% accuracy of the molecules been formed. This is all totally a chemical data and commercial chemical data only. I think that I have brought a chemical PPT towards you. As I am a medicinal chemist, I have been very obsessed with the chemistry itself. So I have been showing you the molecules only. The biological side of the molecule is actually what you can explore by yourself by giving the drug to the poultry itself. We are open to that, and we can give the samplings and drugs to you. You can check by yourself. You can add it to the regular system, and you can say that how it is working for you. Any kind of questioning which you will be having on that antibiotic, we will be available for you. Me and Dr. Subhranshu will be answering any questions you will be having regarding to the antibiotic. So these all are the synthetic process which we are doing for the drug. And what you see here is that the efficiency of production also. Majorly what I have kept it into this slide is that when you are developing an antibiotic, the major source is that can you provide the sustainable amount to the industry or not. Means if the capacity is not there to produce the antibiotic, if you are giving me the orders of 100 ton, then will I be able to compete with that? That totally depends on how much step I am taking to synthesize that molecule. If there are 12 steps, then definitely that product will be more expensive and it will be creating a lesser amount than the existing drugs which have been there. In animal husbandry, you may need a drug which is easily synthesized. You can form it into the daily process, and also it is effective. Right now, many human drugs are there which are cheaper actually. But the thing is that they are cheaper for humans only. When you take to the animal husbandry, there are 7,000 to 8,000 rupees per kg, which is not affordable into the animal husbandries. But they are very good antibiotics, which you can use. So major issue till now, right now, is this only, that there is no one or no product which is available in a cheaper price, which can give you the good results also, plus it is affordable.
Mr. Punit, you only have 5 minutes to wrap up.
Okay, yeah. So I think I should wrap up. Any questioning you have, you can contact me directly. Yeah, this is my mail and number here. You can take a photo and contact me for any kind of questioning, and if you want any samples for antibiotic, we can provide them till if it is 100 gram, 200 gram. If you will ask for 100 kg, it will be a paid one. Right? Thank you for everybody for inviting me here. And I will say you should take a big round of applause for Dr. Subhranshu here. Please, sir, have a stand. Thank you.